Yeah. Hello everyone. This is Buddhabhushan. I recently defended my PhD thesis. So today I will very interesting species that is extinction icon thylacine. So title of Illuminating the Mystery of Thylacine Extinction, a role for relay selection and gene loss. We all know that thylacine is like a species, although it looks like carnivore species, but it is actual marsupial species, and it is as Tasmanian tiger because of it has stripes-like patterns and wolf because of jaw. And it used to be found in mainland of Australia, but and New Guinea around 3,000 years ago, and it was restricted to the Guinea. And again in Tasmania, it used branded as a ship killer, and therefore government introduced the bounty system one and ultimately that last thylacine, which was named as that died on 7th September 1936, and that day now marks as a extinction day. So, this is extinction. So this is very interesting species and much more focused because of trying to de-extinct or genome editing. They are bringing this animal back. So to do this species was there in museum samples. So from there, which was around 110-year-old, they extracted DNA, they sequenced its genome. So this is the sample from where they extracted the DNA, ancient DNA. Their focus was like building genome assembly, what is its phylogenetic, what is its demographic history in past years. They also focus on repeat landscape and diversity. So our goal was like this previous study focused on demographic history and what is the current official convergence with the candidate. So this makes the resource able to study gene losses. So for example, genome assembly was there in 2000, then again chromosomal assembly in 2022, then palindrome 2023. This allows to do look at the gene losses which are specific to thylacine. We leverage this opportunity and we screen what are the gene losses. So why to look gene losses in thylacine? In that previous studies reported that when the genes lost from the species, they are compromised with the immunity. For example, the case of PON1. So when it is lost, so these marine mammals compromise their immunity to defense against organophosphate compounds. Again, further, again, there is one receptor, CNR2. When it lost parrots, they become susceptible to neural inflammation. Again, very known antiviral genes, MX1 and MX2, when they are lost from the tooth wells, they are become susceptible to virus diseases. So now it's clear that like when gene is lost from species, generally it's seen as adaptive evolution because blocks can be compensated by losing one or two genes, but when they are lost, can also be detrimental for the species. So to keeping that in mind, so we decided to compare thylacine versus devil. So we use TOGA, which is machine learning based tool, which identifies candidate gene loss, blast in search in the shorted database, and to confirm that whether are truly lost or not. Then again, this data. So there was challenges like there was low coverage. Again, there is no. It is just based on the Illumina sequencing. So to qualify as a gene is as we mark there should be no gene frame disruption and override should be intact. All gene loss when there was multiple events like frame disrupting. I will talk about in my and we also check whether it is high or not. Then we was like mark as unclear, less than 5 reads short read by supporting, and GC content 55%, and like multiple exons are missing or exon deletion because of this could be artifact short readings. To like upon doing this study, what we found this has lost this 4 specific gene class like CUSD1, HSD17, VD79L. So and again we check are lost across the marsupial phylogeny or they are restricted to thylacine only. Like you see here, the red marks it is lost and the cyan color indicates that it is intact. See, these 4 genes are specifically lost to the thylacine. And interesting is like Sambian, which is only lost in the thylacine, and with other species it is intact. So how we confirm how they are lost? So we check and overlap. So compared to the Devin, there was A, but A base got deleted in the thylacine. Again, let's say, for example, here there is TA in frame stop codon. For example, that C got converted to T, it makes TA, that is again in frame stop codon, that premature termination of this translation and making SAMD9 L in thylacine. Like this, there was GGTG, which again and making frame disruption. Like this, we confirm mutations across other genes like HSD17, B13, BW7. There were multiple frame disrupting changes that led into the surigenation of this gene. So these are the evidence for each frame disruption by the short-read data. For example, there was splice site mutation, like there should be GT. Thylacine it become GA. So that splice site changing again causing that surigenation. Apart from that, there is one base deletion. Let's say in the devil there was G base. G got deleted in the thylacine. Same example, if you see the case of in frame stop codon, there was TAG. In the reference species, it was TAC. Reference species, like in devil, it was TAC. It got to TAG, and leading to similar for VW7. So another thing what we did, we estimated what time they got lost based on the molecular clock. So what we found, they were all lost after this middle Miocene climate. So it is around 15 to 13 million years ago, which is shown here yellow. So if you see the timing of these all gene losses, they are after this MMC time period. And what exactly is reported, this thylacinidae were small-bodied and generally found until this MMCT time period. After this time period, they become hypercarnivorous and the body size got increased. And this is the reference for this study. Again, we check whether like SAMD9 was interesting. So as it is lost thylacine only, we check across the mammals. So what interesting thing we found? So in horses, which are herbivorous, so there this gene was intact. So here green color shows it is intact and red shows it is lost. And if you see the case of all carnivore species, which is shown here in the red highlight, there it is lost. So it is interesting that this all carnivore species lost the gene. Then again, what is the probability of SAMD9 gene being intactness with the of endothermic vertebrate in diet? So what we saw, the probability of being intact decreases by increasing the percent of endothermic vertebrates in their diet. Then, and this gene losses has been seen during transitions like single cellularity to multicellularity, again from water transitions. So these all genes have immune functions. So we strengthen that like this loss could have like harmful to previous cases of the gene losses in the marine mammals. Then the SAMD9, it acts as a restriction factor. So when it is involved active, so it marks that like indicates that thylacine may have compromised immunity. Again, we check whether these genes are expressed in comparative. So we found that these genes are expressed in comparable tissues. Then what we found, there were multiple olfactory receptors, for example, which is purple boxes. So they shows relaxation signatures. And to see here, so why this relaxation signatures, the potential reason could be like, for example, if you see devil, it is small bodied and it is obvious that it will be rely on sense of smell rather than the sense of vision. If you see the devil, it is larger increase in body size, it will definitely rely on the sense of vision than the sense of smell. So this has been reported that people have anatomically studied, like they have found that thylacine has smaller olfactory lobes to the devil. To conclude that, so what we found, there were like SAMD9L, HSD17, V13, CUS-D1 and V, they are lost around after MMC time period. To see that there is pervasive loss of olfactory receptors in thylacine and in the relaxation signatures. The timing of this gene loss, like after the MMC, like to be specific, they are lost around 13 to 1 million series. And this is the time period when this thylacine become hyper and got increase in the body size. Then we see that associated with the carnivore since it is lost in the all carnivore species. And genes are identified. So these are extinct genes. So helpful in the de-extinction efforts of the thylacine. So this last year in the Royal Society Proceedings B. So you can see this study. With this, I would like to thank my supervisor, Dr. Nagarjun Vijay, Krishnamik Kunte, my thesis committee members, also like SSC for International Travel Award and Evolution 2026 for this virtual conference and opportunity. Also like to thank my institute, ISA Bhopal and CSR for funding. With this, thank you for listening.
Thank you, Buddha, for a nice presentation. Yeah, please, if you have a question, you can raise up your hand or ask in the chat box, and you'll be given the opportunity to ask your question. Okay, there's a question in the chat box. Mark, could you please help us read it out?
Oh, sure.
Anna,
Oh, go ahead.
Anna asked like similar patterns of specific lineages gene loss in other specialized carnivores are thylacine unusual. Yeah, like as I mentioned that the SAMD9 was lost in thylacine and it is concurrently lost in all carnivore species while being in. So yes, we can find this pattern.
I actually have a question if others don't. A real quick one. Fascinating talk, amazing work. I'm so curious. So I see that you were comparing the thylacine or showing the Tasmanian devil. Are there other carnivorous marsupials that you know of that could also be used as a comparison, or is that the closest one currently? Thank you.
No, like nearby species which has like this gene requires the good quality of genome assembly. We checked this was the species which has good quality of genome assembly, and we also 21 species of marsupial, whatever that on that day was like in good quality.
Thank you.
Please feel free to ask or use the chat box if you're having difficulty with your audio. And Akash, please get your slide prepared. Yeah, you'll be the next year.
Okay, sir.
Thank you, everyone.
Is it visible and are the slides moving?
It
Um,
is visible, but we can see both you and your slides twice. So if you could
Oh,
yeah, if there's any way to turn off the double window.
I'll stop share. And again, okay. Over the shoulder. Now? Oh,
Still have you also in.
so wait a second.
It's not the worst thing if we have double of you, but if you can just do your slide, see what
yeah.
Okay. I'll just keep the content only.
Okay.
Perfect. Let's
Okay.
see.
Is it fine?
Yeah.
Yes.
Okay. Okay. Thank you.
Yeah.
Thank you.
Sorry,
I'm happy
before you start,
Huh?
can you please enter presenter mode?
क्या?
Your full screen. Yeah.
Oh, you want
Yeah.
it?
Yeah.
Okay.
Yeah, nice. Yeah, so I'll quickly introduce you before you start. Yeah. So welcome Akash. Yeah, so
I'm Akash. I'm currently a faculty at VFSTR Hyderabad. So I'm a computational evolutionary biologist. So when should I start? Shall I start off? I think
Yeah, please
time
go
has
ahead.
started.
Please go ahead. Yeah.
Okay, so I'm happy to have you all here. So let me drive through the work which I have done in 2025 on insect macroevolution. I'll not try to go into nitty details. I'll try to finish on my presentation first so that we can have some more time for question answer sessions. Okay, so basically what I'm trying to do here is I'm trying to answer an ecological question with help of evolutionary logic. With the statistical evolutionary logic, insects are one of the most diverse and successful organisms. Okay, you see them you see the huge diversity in their clades. What kind of can a study of their macroevolution give some insights to their ecological success? Okay, and if I talk about the traits, there are morphological traits, there are genomic traits, and morphological traits and genomic traits, we are by default we are not expecting them to behave same. And how these differences in their pattern, how can it give some insights into their ecological diversity and ecological success, and is evolution being evolutionary close can also give can being evolutionary close also mean that you can get similar patterns of macroevolution? These kind of questions we are trying to see. Okay, so we have taken morphological traits. We'll all from public database only. We have taken head width, head height, head length, thorax width, forewing length, body length, bite force, and then genomic traits also we have taken from the NCBI based website, and species richness we took from the literature, and then we constructed time calibrated trees, and then we try to employ rigorous methods because we try to see that sometimes simple matching of the signal, simple matching of the trait evolution sometimes did not give the results which we are trying to look for. Sometimes some of the analysis were missed. So what we put up a rigorous framework here. First one will be more basic phylogenetic comparative methods. Like we'll try to see what is a phylogenetic signal. Okay, whether that trait in that order, is it having some phylogenetic signal or not? And then fitting it with the classical models. Okay, and then trying to see the correlations with the help of both PGLS as well as phylogenetic mixed models. And then trying to see how it associates with diversity and how it associates with the evolutionary distance. And then we'll later on move on to detecting the pulses in the evolution, pulses in its that traits evolution, like whether the trait did it jump as it was evolving during speciation phases and all, did it show some burst in that trait diversification, so that how it deviated. And then after, okay, if it is deviating, what kind of model it is fitting? Is it a single jump, double jump, three jump, or is between this jump integrated with Brownian motion? Is it explaining the trait? Like that kind of questions we try to explore. And when, okay, now we have to compare the signal patterns or comparative whatever metrics we are using, we have to compare with the evolutionary distance. For this, we needed a background tree. We made a background tree with custom R programs. Okay. And then we try to quantify the tempo and mode that can do these morphological and genomic features. Are they having some distinct more tempos? And is the phylogenetic proximity also means similarity in evolutionary constraints, like that? And how these pulse evolution, how it is going to explain the insect diversification? These kind of things we are trying to see. Okay. So morphological traits, as we expected, because most of the morphological traits, they will be under developmental controls. So this developmental controls also means that there is a signal. But we see that not all the traits are having signals. Not all the traits are having signals. There are certain traits where there is lack of signals. So there is like bite force. So it is allowing the insect to colonize nearby niche. Bite force will be something which an insect will need to defend itself or to forage. So there are some traits which are labile. And then for genomic features also, we see that GC skew is labile, chromosome number it was unexpected. It was also somewhat labile. Gene counts and all, some of these places features were also having low sample size. So gene counts, some of the traits have low sample size, so that has to be seen with care. And genome size and GC content, as expected, they were showing strong signals, strong phylogenetic signal. Okay, so this kind of thing was also reported for microbe also. Okay, so like that. And then we try to see how this, if I were to just compare the signal and try to correlate with the signal with the species richness and correlate the signal with the evolutionary distance, I am not seeing any correlation, neither with richness nor with evolutionary rate. So simple comparison of the signal, I am not seeing any correlation. Okay, then this is showing the signal only. So I'll try to first wind up the thing, then we can go for more details. And then trait evolutionary models for morphological modes, we try to see that most stabilizing models are there. Stabilizing selections or Brownian motion models are predominating for morphological modes. And bite force, which is not showing signal, it is showing white modes of signal of white trait evolutionary modes. And genomic size and GC content, these genomic features, wherever there are these chromosome numbers and all gene counts, whichever were not showing previously signals, they were showing also white modes, white models. The genomic traits which were showing signals, strong signals, they tried to show trait models which are consistent with pulsed evolution, like kappa model, early burst model, with so these with kappa values with closer to 1, they try to show more closer to 0. Sorry, they show that there is a tendency that this lineage, this trait is showing pulses in evolution. And then we tried to, these are the simulated values which we try to see. And then we try to see how these traits are integrating with each other. Okay, morphological traits with applied with phylogenetic corrections, we are seeing them integrate the head traits, wing lengths, they're strongly correlated. And bite force, which did not show any correlation in which did not show any signal in our previous analysis, which showed white models in its phylogenetic evolution. Bite force is also showing weak integration with other morphological features. So it is again supporting that this bite force is more labile feature. And it's allowing the insect to colonize nearby niche. Okay, and then we tried to see that genome size and GC content are correlated. Predominantly, there is negative relationship, and GC skew and genome size are correlated, but that slope is near 0, which means again genomes GC skews not having the changes in genome size is not seeing it's not accompanied by changes in the genomic composition in terms of GC skew. And then as expected, these relationships tend to be lineage specific. And then we tried to see, okay, which features are going to be most important in developing, okay, wing length and head wing length and is known to be correlated with other morphological features. Which of these features are strongly correlating? And we find that for most of the clades, we are seeing the strong correlation with head features, head morphological features, thorax, head width, head height, and all that. Okay, so these are the importance coefficients for, I've just given, for example, I've taken 2 orders, I've kept there. So, and then this, then we try to see here that the work is trying to show that morphological features are showing gradual changes in their trait values, This is somewhat logical also because morphological features, they are under developmental constraints. So developmental constraints is not going to allow for punctuated evolution. Genomic features, they are under they are not under this kind of developmental constraint. So there is a possibility of punctuated evolution here. Okay, so we try to see the morphological features and we try to compare with KS test and AD test. So we are seeing that we are seeing no significant deviations. Okay, and then when we try to see the same thing with genomic features, we see that for the features which we have seen previously significant signals, we are seeing deviations with respect to KS and AD test. Okay, so now, so we try to see that morphological pulses are mostly rare. And these morphological, I did not say that they are totally absent. There are some lineages which are showing morphological pulses. So some of those features, when we try to see, they were odonata. Odonata is one of the most species poor trait in this species poor trait in the lineages which we have studied. So this is indicating that this morphological pulses could have been maladaptive. And then genomic pulses, they are very pervasive. And the feature and the lineages which show genomic strong large jumps in their genomic features, they are species-rich lineages like Coleoptera, Diptera, Hymenoptera. They are showing strong genomic. They are showing strong pulses in genomic features, which may be indicating that these pulses these genomic pulses could have been may have something to do with the species that lineage evolutionary success. Okay, so I'm slowly running out of time. So we can see that species poor clades are they tend to show pulses in their morphological features, which can be which could be explained by maladaptation, and then this pulses species-rich clades are showing pulses in their genomic genomic traits. Okay, so this is this what we are trying to we try to see that this what we try to see that insect macroevolution is characterized by morphological stasis and genomic pulse genomic evolution. So evolutionary tempos are trait dependent, and then these pulses have the pulses, depending on whether they are in morphological traits or whether they are on genomic traits, they have contrasting evolutionary outcomes. So this is the work of this is available in as preprint, and the data are available in Zenodo. Okay, so thank you. Any questions?
Thank you so much, Akash, for the wonderful presentation. Yeah, so please, if you have any question, you can raise your hand, unmute yourself, and ask your question, or you can use the chat box. Yeah, Anna, please go ahead.
Hi, sorry, I was trying to understand how to raise the hand, and that's why I was writing on the chat. So I'm not sure if I understood. Thanks for the talk. Not sure if I understood. Were these pulses more concentrated near the major splits?
I didn't understand, ma'am. These morphological.
These evolutionary pulses that you were talking, were they near? So they were more concentrated near the major phylogenetic splits.
Ma'am, this ma'am, I don't know about the split aspect, split per se, but I can see the pulses. So this says that those traits for that lineage followed a pulse motion. So it is
Okay.
those pulses are generally occurring at the time of lineage diversification. At the time of speciation,
Okay.
we generally see that jump in values.
Okay. Thank you.
Yes, ma'am. I can see another question with bite force
So
specifically. Have you
yeah, so
considered?
Akash, you can reply to that person in the chat. Yeah.
Okay.
Yeah,
Okay.
and now you can share your screen. Yeah, thank you.
Okay.
Okay. Share this. Okay. Is it good?
Yeah, you're good to go. Yeah, welcome,
Okay.
Anna. So Anna will be presenting from mainland to islands, how environment shapes plumage darkness. Anna, you have the floor.
Thank you very much. Good day to everyone, and thank you for being here. My name is Ana Leitão. I'm a researcher at CBU Biopolis in Portugal. Today I'll be talking about part of a project where I'm studying the evolution of plumage coloration on island birds. And today I will talk to you about island and mainland differences in bird plumage darkness, and specifically what are the factors that drive these differences. So first, why islands? Islands have long been considered powerful natural laboratories for studying evolution because they are geographically isolated and they often environmentally simplified. So they provide repeated opportunities to observe evolutionary divergence following island colonization. And this has led to the concept of island syndrome, describing repeated shifts in traits in island species, often markedly different from their continental ancestors, in traits such as morphology, behavior, and life history. So there are many examples, body size differences, morphology differences in the bill, or animals that are tamer on islands. And one frequently reported pattern is the evolution of darker colors in insular populations. So variation in plumage darkness, because I'm talking about birds, or achromatic coloration reflects largely on differences in the deposition of pigments, often melanin, in the feathers. Examples of darker island forms in birds include the chestnut-bellied monarchs, the figure that you see on the top right. We can find them in the Solomon Islands, the white-winged fairy wrens, subspecies that are dark on an island, while in Australian mainland they are blue, or the veiled black-cap morph of the Eurasian blackcaps found in the Micronesian islands. So there are many examples, but these are just a few. And so this raises the question, is darker plumage really a general feature of island syndrome? Several ecological mechanisms have been proposed to explain geographic variation in pigmentation. Two rules are particularly relevant here. You probably heard about them. The first is the Glogger rule, which predicts darker coloration in humid environments. And the potential mechanism include increased resistance to microbial degradation and feather abrasion in these humid conditions. The second is the Bogart rule, or the thermal melanism hypothesis, which predicts darker coloration in colder environments and lighter in warmer environments, because darker surfaces absorb more solar radiation. So it's more advantageous to be cooler in warmer conditions, and the opposite, warmer in colder environments. Islands are generally cooler and wetter than nearby mainland areas at the same latitudes, offering good opportunities to test these effects. So in this study, we asked three main questions. Are island birds darker than mainland relatives? Can climate variables explain these differences, and do females and males differ? And based on previous work, we predicted that darker plumage on islands, wetter in wetter environments, and possible temperature effects and sex differences. So we will go on this now. To test these questions, we assembled a global data set of endemic island birds and their closest mainland relatives based on published phylogenetic information in data sets that we already published. Plumage lightness and darkness was quantified from museum specimens using UV and visible photography, also spectrophotometry, and avian vision modeling processed using PAVO package in R. And the measurements were taken separately for males and females. And at the end, we assembled a data set with color data for 447 island endemic pair species. Endemics and their mainland ancestors. For each species, we extracted climate information, including annual temperature, annual cumulative precipitation, also latitude and body mass. And we analyzed this data using Bayesian phylogenetic mix models, and our models controlled for the phylogenetic relatedness among species, also for the island-mainland pair identity. And we built two models, a simple model where we tested darkness and the predictors insularity, sex, and latitude. And then we included further predictors to test if climate and body mass were related with this. For the sake of time, I'm not going for each predictor and the expected predictions, but I'm happy to answer any questions at the end. So going for the results, we saw firstly that indeed island birds are darker than their mainland relatives. And so here on the right, you see two plots. At the top is a forest plot, and we see that insularity and sex is not overlapping zero, the estimates. And on the bottom, you see a plot for mainland and island in darkness. So lower values are increased darkness. And we see that mainland birds are less dark than island birds. So this is consistent with previous global patterns from Dautrelin in 2016, but here we see it with the double of the data set, of the sample size, sorry. We also found a strong effect of sex. So across species, males were darker than females, and this pattern is consistent with previous work on avian sexual dichromatism. And these differences likely reflect the combined influence of sexual selection, social signaling, and ecological constraints, such as camouflage in females. But now, the most interesting part of this was when we included climatic and biological predictors, the effect of insularity became much weaker. So you see on the forest plot here, the top variable is insularity. So now it's overlapping zero when we included the climatic variables, and now precipitation and the quadratic effect of temperature are significant, so they are not overlapping zero. So climate explains much of the difference in insularity. And going for the precipitation, we found that darker plumage is associated with higher rainfall. You can see on this plot here that the negative effect represents increase in plumage darkness with increases of the explanatory variable. So in other words, species living in wetter environments tend to be dark. And this result strongly supports the Glogger rule and is consistent with findings from global passerines, Australian birds, and parrots. And, as I said before, several mechanisms may explain this association, including the enhanced resistance of melanin-rich feathers to microbial degradation in these humid environments. Temperature also played an important role, not as strong as rainfall, but in a more complex way. So rather than a simple linear trend, we found a non-linear U-shaped relationship. And in this plot, you cannot see the U-shaped tendency here, but birds tend to be darker at intermediate temperatures and lighter towards both colder and warmer temperatures. So this suggests that simple expectations from these ecogeographical rules may not fully capture pigmentation patterns across these broad climatic gradients, and instead multiple selective pressures are probably operating simultaneously. So to conclude, we found that island birds are generally darker than mainland relatives, and this pattern is explained by precipitation and nonlinear temperature effects. And we also identified a strong effect of sex, with males being darker than females. And overall, our results support this idea that increased darkness can be part of the island syndrome, and this pattern emerges from underlying climatic gradients. And that's it. If you'd like to learn more about our team, ongoing projects, and we are always seeking for collaborations, and we have student openings, please visit our website. I'll leave a little bit here. The window opens for you to check it out. Thank you very much, and I'll be happy to take questions.
Thank you so much, Anne, for your presentation. Yeah, so please feel free to raise your hand and ask your question, or you can use the chat box.
Trying to see the window. Yeah, I think to see. Okay. Thank you all. I'll be around it until the end of the session. So if you have sometimes, there's a question. I
Yeah.
can see a raised hand. Yes, please.
Or Rich, you can continue. Yeah.
Yes. Hello. So I was wondering, since you basically argued that most of the plumage darkening is an effect of climate, are there any, any research that test this like on the mainland? Because assuming, for example, with the humidity causes a darker plumage, you should also be able to see that in a continental setting in more humid regions compared to drier ones.
Thank you. Yes, there are some, not specifically on humidity, but yeah, in generally on precipitation and temperature, especially temperature. There's some studies with in the Palearctic birds in the Iberian Peninsula as well that have shown this effect, just not on islands. Specifically.
Okay, thank you very much.
Thank you for your question. Isabella?
Oh, hello. This was
Hi.
very interesting. I wonder because this is a pattern in birds, of course, but in lizards, for example, is the total opposite. But maybe because not so much environmental pressures, but because of competition, stuff like that, they are so colorful. So I wonder in which groups you can maybe extrapolate these findings. Maybe in mammals it will be so similar, but how the multidimensionality of what an island offers shape the color of so many groups differently. Have you thought about this?
Yes. Actually, so on there are two lines here. One is the lightness and darkness achromatic part of the color, and the other one is the hue saturation color part. And birds are also less colorful on islands, but that's another work. I did find a few lizard species, especially in the Mediterranean Sea, that are darker on islands than on mainland. But there's a really strong effect of competition, it seems, also for these darker colors of lizards on islands. Because sometimes melanin patches and melanin in general are badges of status for competition. I haven't thought much about the exceptions, but there are a few things that are working out on these exceptions, and one is the competition and the size of the island that we didn't explore here. Yeah. Not sure if I answer all the question, Isabella, but yeah, it's more food for thought, I guess.
Yes, thank you. But yeah, I was thinking on the Caribbean anoles, for example, that they are so colorful in comparison with the ones in the mainland in the Americas. But yeah, it's interesting to see that in the Mediterranean, something totally different is going on. Thank you.
Yeah, thank you. Okay, again, if you have any questions, I'll be around until the end, and I can answer in the chat. Thank you.
All right, thank you so much. Yeah, Maya, I think You can go ahead. Yeah.
Yeah. For our next speaker, Vidya Kumar, are you there?
Yeah, I am there.
Okay.